Okay, so I did grade that last exam, exam 3. I just finished grading it. So you should be able to see your grade on Moodle. And as I said during the last class meeting, your like course grade should be accurate. Like I said, sometimes there's subtle like little calculations things that get a little messed up on Moodle. And I always double check them. So I'll double check them again, obviously, before I put in your grades. The other thing is that final exam, originally and I sent an email about this, but originally I was going to give you guys, through the weekend to do it, but they're shutting down Moodle as of 4 p.m. tomorrow. So the final exam needs to be finished by 4 p.m. tomorrow. Anything else that you need to hand in, if you're missing an assignment or something like that, all needs to be in by 4 p.m. tomorrow. I think for the most part, we all have handed in pretty much everything. So just so you know, if there is some issue or something happens or whatever, then just email me. But Moodle will be down all through the weekend, okay? And then grades are due on Monday. So really everything needs to be in by Friday. But like I said, if there's some crazy circumstance or something comes up, then please send me an email right away. And so that's the story with that. Are there any questions with that, guys? No? And so remember, I drop one exam grade that does not include the final exam, just so you know. So like if you did really well on all the exams, you're like, I don't have to study for the final. That's not true, okay? So Moodle is coded that way. So from exams 1 through 3, the lowest grade will be dropped, but the final exam does not count in that drop grade, okay? But it's still the average of the exams, and the final counts as 60% of your grade. That doesn't change the percentage or anything like that. It's just that I don't drop the final. All right, so let's review. Let me just pull up the review sheet real quick, and then I guess I'll go over the chapter 12 quiz and the homeworks like I usually do. If there's something else that I feel like is important to kind of bring up, I'll do that as well. I have the PowerPoint sort of ready to go. Does anyone have a specific question from chapters 12, 15, or 16 before I start so that I make sure I address anything? No? Okay. All right, let me share my screen. Okay, so let me first just, again, so the homeworks were due today, the final exam review sheet, the completed final exam review sheet was due. Make sure, if you haven't handed that in, I didn't even check to see who handed in just yet. Make sure that's all in, like I said, by the latest tomorrow. And then that's it. Anything that's not submitted by 4 p.m. tomorrow is just going to be a 0, and I'm going to be calculating your grades. Okay. So like I said, if there's some extenuating circumstance or something that comes up, you need to email me right away because grades are going in on Monday. All right. Sorry for it's kind of short and compact, and the next session, summer session classes start on Monday. So that's why there's sort of a tight time frame there. Okay. So in terms of this final exam, right, it's chapter 12, and that was a pretty bulky chapter. For the experiments here, nobody needs to memorize how the experiments were done. In fact, I already have the exam up. I don't ask many questions about these experiments, so don't spend a lot of time studying that part, okay, guys? And then these are the other topics that are there. Composition of DNA, which we've already kind of talked about before a little bit. Chargraph's rules, there's definitely a question or two involving that. The idea that if you look at the composition of DNA, right, and you look at like the percentage of A and T, right, obviously you would always have the same percentage of A as you do T. You'll have the same percentage of G as you do C, right? Because of complementary base pairing. So it's that idea. Basics of DNA replication. There is a figure. So while I'm going through this, I can just pull up things that I think are important. And I think I point this out in the recorded lecture as well. One second. I could just go to it in a much more efficient way, but apparently I'd rather do this. This figure here, okay, I mentioned this in the recorded lecture that I may show you this figure and maybe you know, block out some of these and ask you, you know, to tell me what, you know, that this protein is, right? For instance, if I, you know, blocked out here where it says DNA polymerase, I might ask you, tell me what enzyme that is or what protein that is and what the function is, okay? But you'll have this diagram to kind of help out rather than just a blind memorization of those proteins slash enzymes that act at the replication fork, right? So these are all examples of proteins and enzymes that are necessary, right, for replication to occur, okay? And each one of them has their own specific job. The diagram helps you sort of remember what they're doing, right? So again, I don't like you guys to kind of, you know, some things you have to kind of memorize, but I try to minimize that as much as possible. Okay, so that's for DNA replication. And then let me go back to here, back to the review sheet. And then obviously we talked about transcription and translation as well. So for transcription, you know, there's a whole bunch of other proteins that are involved as well, but we really didn't talk about that. So in terms of transcription, the main enzyme involved is RNA polymerase, right? That's probably something you should be, you know, have written in your notes there. And then after that primary mRNA transcript is made, it has to be processed, remember, and in those 3 ways. And then, you know what, let me just finish this off and then I'll just pull up one more figure. And then the last thing that I talked about in chapter 12 was translation. And remember, we divided up translation in sort of 3 stages, right? Initiation, elongation, and termination. Also kind of within this, right, we talked about the genetic code and the codons that exist in the mRNA, right? The tRNA has that anticodon and the amino acid on there. So there's a lot of sort of information that probably needs to be filled in this sort of skeleton outline here. All right, so let me just pull up one more figure to kind of review that. And then you guys can let me know if you have other questions. Right, so again, one thing about the genetic code, as I'm going through this, okay, if we look at the genetic code, remember, these are the codons that are in the mRNA, okay? tRNA has an anticodon, which is complementary to these, you know, to the codon, and then an amino acid on the other side. So the tRNA is really sort of the translator from mRNA to protein, right, to amino acid. Also remember some, you know, sort of characteristics of this genetic code in that, you know, for any one given amino acid, there may be several codons that code for it, right? So for instance, if you look at leucine here, right, you can see all of these different codons code for leucine, okay? Remember, there are 64 possible codons. There are only 20 amino acids, okay? I talk about that in the recording. Also remember, within this genetic code, there are stop codons, and there's also a start codon as well. So they don't all necessarily code for amino acids. All right. So if we look at translation, okay, remember those 3 stages that I mentioned. So here again, here's the example of showing you the codon. Sorry, we didn't get to translation yet. Right, the codon, we're just about at translation now. So the codon in the mRNA, anticodon in the tRNA, and then the corresponding leucine at the top, the corresponding amino acid, okay? So now if we look at, right, the best way to kind of study the translation is to look at these where it's broken down in initiation, elongation, and then termination. And honestly, you really just need to know the basics in terms of what's happening. In elongation, obviously you have, the reason why it's called elongation is because that polypeptide chain is getting longer, right? And so basically, you know, the whole ribosomal assembly is there, the mRNA is there. Remember, you have these 3 sites, right, that are on the ribosome that basically take in the tRNA molecules, right, and provide a position for each one. And so you have right here the A site, the P site, and the E site. Okay, and so basically they sort of translocate along. Once it gets to the E site, it's not going to have its amino acid anymore because it donated it to the polypeptide chain and then it gets released. Okay, but so you can see all of that in the elongation part. Sorry, I kind of went backwards. Initiation, remember, it starts with the small ribosomal subunit binding the mRNA. Then the large subunit comes in and everything assembles together, right? Once this start tRNA here, this methionine, the initiator tRNA is in the P site, then it will start, okay? So here's your start codon. And then again, like I said, you can go to elongation, but I'm not gonna ask you like super specific questions about the process of elongation, okay? Even like in terms of like, moving from the A site to the P site to the E site or anything like that, I'm not going to really ask you specifics about that, okay? So don't spend so much time kind of looking at the specifics of elongation and knowing exactly how those tRNAs are moving along within the ribosomal sites there, okay? All right, and then termination though, again, basics of termination, right? It's that basically you're gonna reach a stop codon, right? Once this stop codon gets into this A site here, there is not gonna be a tRNA that binds, right? Something called a release factor binds, and then that just causes the whole thing to disassemble, right? And that polypeptide chain to be released. So we just really wanna know what the main function is of each stage of translation, meaning initiation, elongation, and termination, okay? I did tell you that this figure is handy in terms of kind of just reminding you of where things are happening, right? So remember, transcription occurs in the nucleus. DNA does not leave the nucleus, right? It's in the nucleus. Transcription happens here. That mRNA gets processed, and then it's released, and it goes and finds the ribosome, and translation will occur, okay? Are there any other questions about that? What I will do also is go over that chapter 12 quiz, just as little. I probably reviewed most of the things that are on that quiz, but we can go over it quickly. Any other questions from chapter 12, guys? All right. So let me pull up that quiz. One second. Chapter 12, where are you? All right, there it is. Okay, in DNA, the nucleotide guanine always pairs with cytosine, right? G with C, A with T. Remember that in RNA, you do not have thymine, right? You have uracil, okay? All right, one of Chargraph's rules states that in a species, the amount of A equals T and the amount of G equals C. Again, because of complementary base pairing. All right, so again, if we go from here, these are kind of really basic questions, right? Adenine and guanine. Remember, that's referring to the purines versus the pyrimines. Remember, a purine always binds to a purine, right? It's never two pyrimines together or two purines together. If we talk about the rungs of the ladder, right, of that DNA, we're talking about a purine being bonded to a pyrimine, right? So this is us up here when we're talking about the structure of DNA, right? So adenine and guanine are purines. Remember, if you look at the structure of the purines, they have a double ring structure. And if you look at the structure of the pyrimines, they have a single ring, okay? So again, remember, a purine always pairs with a purine, right? Purine always pairs with a pyrimine. We always know that A pairs with T, but sometimes maybe you don't think about purine and pyrimine. So I asked you about adenine and guanine. Those guys are purines, Okay, so let me go back. Oh, it's under here. All right. According to the Watson and Crick model of DNA, DNA is a single helix and the strands are parallel. Not true, right? It's a double helix. The strands are anti-parallel, meaning one, right, 5' to 3', and the other one is, sorry. So if you're 5' to 3' here, then it's 5' to 3'. So they're anti-parallel. Okay, so that is false. Okay, the function of DNA replication is to synthesize a new DNA molecule. Remember, for DNA replication, we talked about this, right? During S phase, cells are going to replicate their DNA. So before mitosis or before meiosis, the entire genome needs to be replicated. So in replication, the cell is replicating its entire genome. Remember, that's not true for transcription, right? It's not the entire genome that's getting transcribed. It would only be a specific region of the DNA that gets transcribed. Okay. Super slow. Blank is the main enzyme in DNA replication. It synthesizes the DNA by adding nucleotides to the growing strand. That is DNA polymerase, okay? And I think I do say that in the recording, right? DNA polymerase is the main enzyme that actually, you know, adds nucleotides to that growing strand by complementary base pairing. Obviously, there are a whole bunch of other proteins involved, right? I showed you guys that replication fork, so it does not work alone. All right, in translation, a codon is composed of 3 nucleotides, right? And is part of what? It's the mRNA. So it's this one, right? 3 nucleotides in the mRNA. Remember, the anticodon is also 3 nucleotides, but it's in the tRNA, okay? So don't be confused by codon and anticodon. And when you talk about the genetic code, those are the codons in the mRNA, not the anticodons in the tRNA. All right. mRNA is synthesized from DNA in the process of transcription. Okay, so transcription, we make mRNA from that DNA template. Again, remember, it is not an entire transcription of the entire DNA molecule. It is just one region. It's whatever gene, right, that needs to be just transcribed at the time, right? Whatever protein needs to be made, that's where the transcription is going to occur. All right, transcription occurs where? In the nucleus. Remember, DNA, we know DNA is housed in the nucleus. DNA doesn't leave the nucleus. The mRNA leaves the nucleus after it is made. Well, after it's processed, right? All right, during blank of translation, a stop codon enters the A site of the ribosome. A release factor binds. I just talked about this not that long ago. And that ribosome disassembles. That is termination, okay? And so that is it. I mean, look, I could have made that quiz a lot longer. That was it's pretty basic. But like I said, don't spend a lot of time on the beginning, the introductory stuff where I talk about the discovery of DNA. I think it's important to go through. But like I said, I don't ask you many questions about it. Okay, so I don't want you to spend any time sort of memorizing any of that information. All right, now what I'm going to do is I'm going to go back to that review sheet. And, oh, we might, you know what, let me just check chapter 12 homework just to make sure I probably hit everything here. Yeah. So again, if you just want to talk about basic function, right? Function of DNA replication, replicate the entire genome, right? Make it an entirely new, new DNA molecule, right? And so the other thing to remember about replication is that it's semi-conservative, right? So when you are going through replication and you end up with the new DNA molecule, you're always going to have one parental strand, right? And one newly synthesized strand. So that's what we mean by semi-conservative. So that was one other thing that I didn't review, I guess. So the function of the DNA replication, right, is to make another copy of the DNA, okay? Function of transcription, to produce mRNA from DNA, okay? And again, remember, it's specific. It's only going to be a certain region of the DNA. If we talk about the function of translation, right? It's to make a polypeptide chain, right, from mRNA, or make a protein from mRNA, something like that. I was just really looking for basics here because, again, so that I know you understand big picture, okay? All right. So that was that. So if we move on and we look at the review sheet for chapter 15, right? So now after chapter 12, we switch gears, right? That's our last chapter of sort of molecular biology. And the next two chapters are now involving evolution. And so when you move into Bio 113 or General Biology 2, you start off talking about evolution, talking about different species, right? Taxonomy, and then you sort of move on from there. So those two chapters really set up for moving into that sort of different unit, right? Which is evolution. So chapter 15, you know, in the beginning of chapter 15, obviously talks a lot about Darwin, right? And his voyage and what he saw. Also talks about what were the thoughts and the beliefs at the time of Darwin or just before that, right? So that you can sort of get a little bit of perspective in terms of where he was coming from and what his contemporaries thought. So let me just pull up the review sheet real quick. Right? So again, if you think about what other people thought at the time, there were a few scientists that I mentioned. So Linnaeus, you know, they call him the father of taxonomy. He developed that binomial naming system, but he also believed in something called the fixity of the species, okay? Not natural selection, right? Definitely not close to what Darwin believed, but I think if you want to think of one of his major contributions, it was really developing that naming system. Some other scientists here that, again, were around during Darwin's time, Count Buffon, Descent with Modification. Cuvier is basically founded paleontology, but talked about catastrophism, okay? Lamarck, there's a picture that I showed you guys in the recorded lecture that shows you Lamarck's belief, right? That inheritance of acquired characteristics versus Darwin's natural selection, okay? So let me just pull that up because I think it's kind of important to go over that to make sure we understand that. And then a lot of the rest of the chapter talks about Darwin's theory of evolution, right? Which is based on natural selection, right? And adaptations. We then talk about artificial selection a little bit. And then talk about, so then some other sort of evolutionary changes where we're talking about more modern examples of evolutionary change. And then the last part of the chapter talks about evidence for evolution, right? In terms of, you know, how do we know and what's sort of there to support Darwin's theory of evolution, which is obviously a well-accepted theory in biology, and these are some of the things here. The one thing that obviously is a more recent type of evidence is the biochemical evidence, right? I mean, Darwin didn't have access to that at all, but as technology has improved and we have the ability to analyze DNA and to analyze protein sequence, all of that evidence also supports his theory, right? So it's important to kind of think about that. I mean, obviously, if our biochemical evidence that we have now didn't support Darwin's theory of natural selection, well, you know, there would already be some other theories that were out there, but that's not the case. It does support that as well. Okay, I guess let me just pull up just a few things, and then that's pretty much it. So where are we? Chapter 15. I just wanted to just go over this. So here, so this is the one slide I was talking about, right? If you look at Lamarck's on this side and Darwin's on this side in terms of explaining how giraffes basically ended up with these really long necks, right? If you go according to Lamarck's inheritance of acquired characteristics, the idea is that all of these giraffes early on had short necks, but during their lifespan, their necks were able to stretch to reach those leaves, and then they just gave birth to offspring with long necks, right? Not quite the way it occurs. So if you look at Darwin's on this side, oopsie, I always do that, right? You had variation in the population, right, of this early population of giraffes where some had long necks, some had short. And what happened is that those giraffes with shorter necks ended up dying off, right? Because they were not suited for that environment that had these, you know, leaves that were really high up. And therefore, the ones that survived, the ones with tall necks, were the ones that then produced offspring. Okay, so you already have variation in the population, right? Then the natural selection part, the environment selects for those individuals that are most fit. All right, so I mean, that's natural selection in a nutshell there. So again, talk about sort of this biogeographical evidence, right? Where he saw different species of animals in really different regions of the world, but where he noticed that landscapes were the same, he saw similar animals. Okay, so this idea of putting together environment with what he saw in terms of physical characteristics of the species helped him come up with his theory. All right, and so that's what what we were talking about there. The other thing I just wanted to point out, and so again, these were some more modern day examples of sort of evolution, right? And then the last part was evidence, right? Fossil evidence, we talked about biogeographical evidence, okay? The one example with marsupials, in terms of them being geographically isolated, okay? And so that's why you only see marsupials in certain areas, because they didn't have the competition of those placental mammals that happened in other areas. Okay, and the last thing I just wanted to show you, and so we talked about, I also talked about homologous structures and analogous structures. So the existence of homologous structures, right, like the bones that make up the wing of a bird, the wing of a bat, the arm of a human, they're all similar, indicates similar or common ancestral origin, right? Okay, and supports his theory of evolution. Also, you see lots and lots of similarities in embryos, especially early embryos, okay? And then the last part talks about biochemical evidence, like I was mentioning before, okay? So this graph here basically shows you the number of differences, amino acid differences in cytochrome C, which is a protein that's involved in the electron transport chain. And so if you look at, right, if you look at from the human all the way down to yeast, those species that are more closely related to humans will have a smaller number of amino acid differences, right? Makes sense. So if you look at the human versus the yeast, and between the yeast and the human, you have about 50 amino acids that are different. If you look at it between the monkey and the human, maybe there's only 1 or 2 amino acids that are different, okay? So again, that's an example of biochemical evidence, but like I said, you can also look at DNA sequence and kind of maybe make a similar sort of graph for some gene or something like that, and it would be the same idea, right? Where you see more differences indicates a more distant relatedness, where you see more similarities, they are more related, okay? So I think that that's kind of pretty much mostly what I wanted to point out for chapter 15. This just talks a little bit, again, this is more of this biochemical evidence, particularly in developmental biology, and the idea that genes that are responsible, right, for a specific function during development, you'll see these genes across all species, right? And then you'll see subtle changes based on what type of species you're talking about, but the same gene is responsible for sort of the same function, right? Whether you're talking about a chicken, or a human, right? All right, so that's pretty much that. Let me just check on our what our homework questions were. Chapter 15. I think I probably already addressed them. Go away. All right, so describe the differences between the Lamarck's and Darwin's theories. I explained that, right? That one figure basically is illustrating that, okay? Name 3 observations that natural selection is based on. So it's the idea of, you know, seeing similar species in similar environments, right? Things like that. So you can kind of come up with different examples there. The biochemical evidence that I just discussed, that could be one of them as well. Okay, so the idea that, you know, animals that are more suited for the environment will survive, the other ones will die off. Those are all sort of observations that sort of illustrate natural selection. Okay. You know, and again, kind of, and if you think about any of those things that Darwin observed during his voyage, that was how he came up with that theory, right? So these were all observations that helped him develop that theory. So anything along those lines would work here. Okay. So that's just kind of meant to kind of make you kind of think about, you know, what evidence would essentially correspond to the idea of natural selection. Any questions, guys, from chapter 15? All right, so last chapter is chapter 16, which is smaller scale evolution, right? Or what we call microevolution. So on a population level. Let me pull this guy up first, and then we'll go from there. Why is this always in my way? Okay, so like I said, this chapter really talks about population genetics, right? And that's a branch of biology, right? Population genetics, the idea of just analyzing a population and looking at the number of alleles that are there, right? Allele frequencies or genotype frequencies, meaning, you know, how many dominant alleles are in that population, right? Or how many recessive alleles are in that population. And then if you looked at, say, time 1, right? And then you looked 4 years later, are those allele or genotype frequencies changing? Okay. And so I mentioned and discussed Hardy-Weinberg equilibrium. I want you to kind of just be familiar with the concept. The details of that, again, are a little more advanced for this class. So I like to just introduce it so that you guys can sort of see a mathematical tool that geneticists, that population geneticists would use to analyze whether a population is evolving or not, okay? And maybe it tells you about changes in the environment, right? Maybe some invasive species, pollution, right? Pollution, there are many, many things that can affect a population. You know, you could think of it even within the scope of disease, right? If you were looking at a given population and you were looking for the number or the percentage of disease alleles that are in that population, and you see that in a given 5-year period, all of a sudden that percentage has increased dramatically. Well, something's probably going on, right? Maybe it's something that's induced by some carcinogen, right? And you're talking about cancer. So I think so just to kind of put things in perspective, it may not be about analyzing a moth population. It may be analyzing a specific human population and involving a specific disease. Okay, so important tool that they use. Am I going to ask you specific questions like those P plus Q in the equations? No. Like I said, you go over that in genetics. This is just meant to be an introduction so that when you get to genetics, you're like, oh, hey, I remember that. I understand what the purpose is. Then you can really get into that a little bit more. Okay, so again, don't spend time, you know, really like hemming and hawing over the Hardy-Weinberg equations. Honestly, if you do spend a little bit of time with it, it's not that hard to understand. But for this class, you don't have to worry about it too much. Okay. Again, it's a tool that population geneticists use to indicate whether evolution has occurred, and usually it's within a more shorter time frame, and it's also within just a given population. Okay. The example that I give and that the book gives is this peppered moth population. But like I said, you can think about it with human and a disease allele, okay? Makes it a little more interesting. Okay, talks about though what's necessary for a population to be in Hardy-Weinberg equilibrium, meaning there's nothing changing. In reality, that doesn't happen, right? There's usually always something that's going to be changing and some sort of evolution that's occurring, okay? So there's sort of this table that shows you this is what you need for a population to be in equilibrium, okay? In reality, that doesn't happen, but it's a mathematical model for geneticists to use to see how much something is changing, okay? So just kind of give you a little bit of perspective on that. All right, and then we talked about causes of microevolution, talked about mutations, right? Things like gene flow, genetic drift, bottleneck, founder effect. You should just be familiar with those things there, those types of genetic drift and what have you. Also in that chapter, I talk a little bit about different types of natural selection. Okay, so there was 3 different types: stabilizing, directional, and disruptive. And then at the end of the chapter, I talked a little bit about something called heterozygote advantage. Okay, meaning that if you have one, so if you think about sickle cell, right? And malaria, that's the one example. There's also a cystic fibrosis example I think that I gave as well. But in areas of the world where malaria is prevalent, you see a much higher percentage of the population having the recessive allele for sickle cell, okay? So having one copy of the sickle cell allele protects you from malaria, okay? So that's the idea, that in areas where malaria is prevalent, you're actually protected as a heterozygote for sickle cell. So that gives you an advantage, and that's why it's called heterozygote advantage. Okay, so pretty interesting. Like I said, you'll see an increase in frequency of those recessive sickle cell alleles in areas of the world where malaria is prevalent. Okay, so that's what's meant by that. Let me, and then just, I'll pull up chapter 16 real quick, but and then remember, there's just some additional topics on there. Function of membrane proteins. So like if know the function of a receptor protein or a channel protein, that's what I mean by that. In terms of biomolecules, know the synthesis and degradation. Remember we talked about hydrolysis and dehydration reactions. You should just be able to, you know, recognize and identify those. Biomolecule functions, meaning like functions of lipids, functions of carbohydrates. And then levels of protein organization. Remember the primary, secondary, tertiary, and quaternary structure of proteins. Okay, that's when, you know, you have your polypeptide chain, that's primary, but remember it then forms secondary structures and then folds more before it can be functioning. Okay, so those are those additional topics. There's nothing else additional on there. It's those specific ones there. All right. So let me just, I think there's just one thing I kind of wanted to just pull up for chapter 16, and then we'll go from there. So that's where we're talking about heterozygote advantage, right? So it says maintenance of diversity. Remember we're talking about, right? It's advantageous to maintain diversity. There's a reason why. By keeping that recessive sickle cell allele around, it actually ended up being protective for some individuals, right? If that sickle cell allele went totally away, right, because it you know, it potentially causes sickle cell anemia, you wouldn't have had that advantage, right? So always best to have a diverse set of alleles that exist in a population. All right, this was the only other thing I wanted to pull up. This was the 3 types of natural selection that I was just mentioning, right, in terms of how it sort of changes, right? So depending on the environment, right? So the peak narrows here in, oh, sorry, it would help if I actually, oh shoot, I wonder if I got rid of that. So here, right, you have directional, right? You have stabilizing, intermediate phenotype, and then disruptive, okay? So the first one is directional, right? You have this narrowing of the peak. Here, this one, B, right, is stabilizing, where you get this intermediate phenotype. Oh, yeah. One second. Yeah. Wait, I want to just make sure I didn't screw up these here. Yeah, the narrowing, the shift is directional. Yeah. So stabilizing, directional, and then this one is the disruptive, where you end up with 2 or more, okay? So sorry, stabilizing, right? Directional, you have a shift, okay? It shifts off center here, and then this is 2 peaks where you actually have 2 phenotypes that are favored. So let me just give you an example. Stabilizing example is birth weight, right? That you have this sort of, so if you look at human birth weight, this graph is could be a little confusing, right? But this is, the red is infant mortality, right? So deaths. The blue is percentage of births at certain weights here. So what you can see is that an intermediate range is is more prevalent, also favored because it's going to lead to less mortality. On the extreme ends here, right, being low birth weight or high birth weight, you're going to have a lot of deaths, okay? So when it's stabilizing, basically what's favored is the intermediate phenotype here, right? So you have this narrow peak, because on the extreme ends, those, again, low birth weights and high birth weights are going to end up dying off, okay? Because it's not good to be tiny, but also if you have a really high birth weight, it may indicate some other type of issue, maybe like hydrocephaly or something like that, where it indicates some other type of disease. Directional is where you see this shift in size over time, right? So like initially horse size, body size started off small, but then over time, as the environment changed, this larger body size became more favored, okay? And then for disruptive, depending on what exact area in a species might live in, you could have the basically equal sort of favoring phenotypes, and you end up with sort of these two peaks, okay? So that's what those are showing in terms of the different graphs there. All right, let me just look at the chapter 16 homework, and that's pretty much it. So genetic drift, and then it says, what's the difference between stabilizing and directional? I just went over that, okay? So if we just look up genetic drift here, just to review that, we can. So remember these, what follows these slides here, the genetic drift slides, are examples of genetic drift, okay? So what is genetic drift? Changes in allele frequencies, right, of a population that are due to some chance event, okay? It could be a flood, it could be a tornado, it could be a hurricane, but basically random members of that population are going to be removed, okay? And then obviously that will then change the allele frequencies or change the percentage of alleles that are there, right? Depending on which individuals are removed from that population. Also, if the population is small, genetic drift is going to affect that population a lot more than a larger population, okay? So again, then these are examples, the subsequent slides are examples of genetic drift, okay? Sorry, I don't know why it's not letting me show. So that's what this is showing you here, right? So some natural disaster, here's your little population of frogs, right? Where you got only, the brown frogs are only 10% of the population, right? And so a natural disaster happens, and basically it kills off a whole bunch of green frogs. And so now this brown frog ends up being a much higher percentage of the population. If this brown frog ended up being killed off, you would end up eliminating alleles potentially, right? So again, you can see how genetic drift or something like this will affect a smaller population a lot more than a larger population. And so like I said, different examples of genetic drift, right? Bottleneck effect, founder effect. Okay, these are all different types of genetic drift. All right, I think that that is pretty much everything, okay? So let me just double check that. Yeah, so like I said, all assignments need to be in by 4 p.m. tomorrow. That just negates any issue that might happen with the Moodle, whatever they're doing upgrade. I think it's because we're moving to university status as of July, whatever. So there's changes they need to make to Moodle. So, so anyway, it seems like it's a pretty major sort of update or whatever. So I don't even want to, overlap with that at all. So like I said, if something crazy comes up, you guys need to email me right away. Otherwise, everything's in by 4 o'clock tomorrow. Grades will be in by Monday, okay? By the end of the day, Monday. If there's a question about something, you need to tell me before Monday, because grades are going in Monday, and that's it. And again, everyone can see their grade all along, so there's no secrets. All right, guys, so that's it in terms of review. I hope everything was clearly delivered. I'd rather do this live, but it is what it is. Let me know again if you have any questions or anything like that, but that's pretty much it. So this is our last meeting. Like I said, everything in, grades will be in. If something comes up, please email me right away. All right, guys, enjoy the rest of your summer. Now you can actually get out and do some stuff, right? All right, guys.
You too.
Thank you. Take care, guys.